We're not having a snow day today, so we're back in class, and here we are. And let's just start about catching up with where we're at. So, where we're at is we had 2 snow days. And last Thursday, a week ago, we had a snow day, and I made a relatively short recording just to keep moving the stuff forward. And then on Tuesday, we had a snow day, and I made 2 recordings that were a total time a little bit longer than our regular class meeting, but they all balanced out. So that's where we're at. No one needs to send me their notes on those because we all missed class those days, but they're just as important as any of our class meetings. So you really have to study those carefully, and sorry about snow days, but we just power on in this course. And so where we're at is we have a test in 1 week from today, and it's our first test, and it's going to cover everything up through chapter 17. And so I'm nearly done talking in class about what I want to talk about that will be covered on the first exam. There can be questions, and we could add more knowledge going back to it, but I'll finish the new material that I want to talk about in class today, and then we can talk about anything you want, and we can, if I have time, I'll just start talking about chapter 18, which is cell cycle stuff, and it's very related, but we won't be covering any of chapter 18 for this exam. But if I talk about anything in class, that's connected or that's helping you answer questions, those will be on our exam. Next Tuesday, Riley's going to come, and it's going to fit in really well because Riley is going to talk about the CAR T cell therapy story, and he's going to focus on the receptor, so the CAR receptor and building the CAR receptor, what it is, and then signal transduction pathways that happen in the modified T cell that has the modified synthetic receptor on it. So, what is that about? And he's going to come a total of 3 more times. This next Tuesday, he'll talk about the receptor and signal transduction. A little bit after our first exam, he's going to come in and talking about the cellular response. So the nuts and bolts of the cellular response that these guys make, the T cell makes. And then the 4th time he'll come in is when we're dealing with chapter 20, which gets to really talking about cancer and some therapeutic stuff and things like that. It's still nuts and bolts stuff, and it's just more and more figures like chapter 17, where you're like, oh, here's the normal signal transduction pathway, and this is the gene that's mutated in this cancer, and this is a therapeutic that's targeting that cancer. And so we're already talking about that. On Tuesday in the recordings, I talked a little bit about chemotherapy called cetuximab, and how that works at a molecular level, because modern oncology involves pharmaceuticals that work at really targeting specific molecular pathways that are disrupted. Our super favorite example is Tamoxifen, and how it works, on breast cancers that are ER positive, because it is a estrogen receptor antagonist. So you would need the estrogen receptor for them to be able to effectively slow down a cancer like that. Okay, and so Riley's going to come in, so that'll be stuff. And I don't think Riley is going to have us read anything. And his topic, CAR T cell therapy, is just one figure in chapter 20. So when Riley's talking, it's integrated in our course, and it's helping us understand our course, and there could be specific questions on the exam that comes from Riley's stuff. So Riley might talk about some stuff that if he does on Tuesday, I'll stand up and say, yeah, this is some things Riley said that we really need to know for the first exam. But it's about receptors and signal transduction pathways, so it should fit in fine. And our exam is going to be 52-point questions like that, like what is this, what is that? And you want to start out just being really solid on everything we said in class. Everything that's said in class is considered really important because we have such limited time. And that's one reason I record things, is you really can't miss a minute of class, either by an absence or by just being distracted, during class. You really need a chance to get that learning done. Most of it is also represented in the book like that, and then there'll be a little bit from the book that just never was covered in class, and we've talked about that. So that's where we're at. remember that Riley's a tutor. I don't know that anybody's contacted him yet. That's fine, it's understandable, but he's great, and, take advantage of that. And so that's where we're at, okay? And we can start here and see if people have questions about anything at all. Yes, Hannah?
Tamoxifen is an antagonist, right?
Yep, it's an antagonist. Yep, yeah, yep. And we learned about an agonist. Does anybody remember what agonist of estrogen we learned about?
BPA.
Yeah, BPA, that chemical that's in plastic that everybody's trying to avoid, is an agonist. And there's a good chance you've never heard the word agonist before, but it's nice because you can see it's just the opposite of antagonist. And everybody knows antagonist is your enemy, like that, right? Opposing you. And agonist and antagonist are used in endocrinology very specifically to be molecules that bind to the receptor that they're modulating. And if we don't know that they bind to the receptor, we have to call them a mimic. And so we know that BPA as an agonist of estrogen, we know it binds to the receptor, so it's not simply a mimic. It actually binds the receptor. A mimic could do something like increase or decrease the rate of synthesis of estrogen, and that would mimic the effects of estrogen or lack of estrogen. Yep, yeah. So we run into that terminology. That terminology is really from endocrinology, but that's not a course that's taught at USM. So, we want to learn a little endocrinology in Chapter 17. Chapter 17 starts out talking about endocrinology. Let's just skip to, I got a couple of figures from the book that I haven't shown yet. So it's just the very first figure in the book, and it's about receptors. And what it's showing is what we've talked about, and that is that there's going to be a cell called a source cell that's going to make a molecule that is going to be released from it, or like that. And that molecule we like to call a signaling molecule. All hormones, all neurotransmitters, all cytokines are in that category. And that's cell signaling, where a source cell makes a signal for a target cell. And in Chapter 17, we're really interested in the target cell. We've learned about the target cell in wounds under a scab. It's the skin cell that's the target cell. And in that case, it's PDGF. The source cell in that case is surprising because it's actually a cell in the bone marrow that's creating the platelets that PDGF is packaged inside of. And so there's two terms from that. In signaling pathways, there can be really two stories, and one is called an endocrine signaling, and endocrine is synonymous with hormone, and it means a signaling molecule that spreads throughout the body, throughout the whole organism. That's a hormone or an endocrine factor. And then there are factors that don't spread throughout the whole body. They're local, and they're called paracrine factors. Can anybody think of a paracrine factor that we've learned about? So one paracrine factor we learned about is PDGF, right? It's local. It's only under the scab. So it's our favorite example of a paracrine factor. We don't want it to spread everywhere. We want it to be activating skin cell proliferation, the cell cycle in skin cells, right under the wound to help heal, right? When the scab falls off, there's pink skin there. Some of those are brand new cells. Our skin is always growing and dividing, always replenishing, always getting rubbed off, replenishing. So the cell cycle is going slowly in skin cells under a wound. We want it to speed up, and that's the role of PDGF, platelet-derived growth factor, classic name, right? Like that. And we saw the woman with that type of very rare skin cancer that had a translocation where she started making PDGF in her skin cells, like that. This third term down here is not a third type. It really shouldn't be in this same figure. It's called autocrine signaling, and it just means a cell signals itself. Can anybody think of an example that we've learned about of autocrine signaling?
Would MyoD fit in there?
No. So MyoD is not a signaling molecule. It's not going between cells. It's the cell itself, and that's a really good point because when we use the words signaling factor and transcription factor, they sound similar. They're different. Transcription factors are inside cells, they stay inside cells, they function inside the nucleus. Signaling molecules or signaling factors are outside the cell and go between cells. Yeah, anybody can think of a autocrine response that we've learned about? The woman with skin cancer has an autocrine response, right? She's got that chromosomal translocation, so now, oops, she's making PDGF in the cell that's supposed to be the target, like that. And if you look, like I can literally remember my first year in college in intro biology, when they have this in a textbook in biology ever, they always show these three things: endocrine hormone signaling going throughout the organism, paracrine local signaling, and then I remember it in college because I thought, what is this autocrine signaling? That just seems stupid, right? And useless and different. It's definitely different. The reason that it gets put on in these textbooks like this one is because an endocrinologist says, those are the ones you want to list. And very few people know very much about endocrinology. It's really a challenging field. And so textbook authors just go, yep, good, put in there. It's in every intro bio text. It's in our text like that.
Is like programmed cell death in that autocrine system where the cells telling itself I need to self-terminate?
We're going to learn about that in chapter 19. The answer is no. It's a self-signal. This has got a specific term. Autocrine means that there's a factor that's released from the cell, and then the cell responds to it. And the woman with skin cancer is a classic example of autocrine signaling. It's a type of paracrine signaling in her case, right? It's local signaling. So, significantly, you could have some endocrine signaling that is autocrine signaling and some paracrine signaling that's autocrine signaling. It's no big deal. It's no big deal. I'll just tell you the reason it's here is because the cells that make hormones are often regulated by the hormone they make. And so that's why an endocrinologist would be like, oh, autocrine signaling is epic. It seems weird to us because it's like, why would a cell tell itself something? But it's a feedback loop, and sometimes it's used in the source cells, in the endocrine glands, like the adrenal gland and so forth in our bodies, to respond to their own signal. Sometimes it's a negative feedback. Oh, we should turn this off, like that. We'll talk about that this afternoon in the lab class, because we're studying a gland in the lab class. So, just that, okay? And here's this idea of there's going to be a source cell and a target cell, like that. And then the book is just introducing it. It says, oh, wow, there's a lot of different kinds of hormones, a lot of different kinds of paracrine factors, like that. Just a lot of different kinds. In our course, we're learning about several, like that. PDGF is one, estrogen and testosterone and other molecules that bind to nuclear receptors as another example, like that. And in chapter 17, we have a little list, and I crank through most of that list on the recording on Tuesday, like that, because there are just a huge number of really famous signaling molecules. I would say in chapter 17, we kind of take the signaling molecules for granted, and the idea that some other cell is making them, and what we look at is the target cell, and we look at the receptors, and the signal transduction pathway, and the cell response, like that. So when we see something like this in chapter 17, just don't worry about it. The book describes a lot of signaling molecules at the start of the chapter, and they have a lot of different chemistry. Some signaling molecules are proteins, like PDGF. Some are not. Some are steroids, like estrogen and testosterone. Some are peptides, some are modified amino acids, and so forth. Other very different kind of chemistries, like terpenoids and so forth, can be these signaling factors. A cell has to make them. That's our key thing. A cell's got to make them, and then a cell's going to respond to them. It'll have a receptor. Yeah.
So the entire section of signaling via small, other small molecules, we just kind of.
De-emphasize. So you're required to read it, try to absorb some, learn some of that, right? Because I don't really like to say, oh, just read this section. Read that chapter, right? And learn as much as you can out of it. But as far as our test is concerned, you wouldn't dwell on it. You'd read through it once and get a feeling for it, and then you'd roll on. Yep. Yeah. Yep. Yeah.
So the chapters are 4, 9, 16.
Yeah. So the chapters that we cover in the book are 1, 4, 9, and 17.
Oh, 17.
Yep. Yeah. But then it's everything that we've done. So are those clay model videos and assignment and everything that's happening in class like that. Yep. Yeah. So that was just kind of in response to that question. So other questions. So let me just scroll back here and say, you know, this is what we're up to, and we're catching up because we had to be out on snow days. But other questions people have about anything at all? Yep.
The textbook goes into a lot of detail for the MTOR pathway and the PI3 kinase AKT pathway. How much emphasis should we put on that?
Yeah, so the book goes into the signal transduction pathways in a lot of detail, and there are many. And I think I still have it here on my little chalkboard. Oops, not that. Where that's my drawing. I don't know if you've seen the Tuesday video yet, but I'll recreate this. And so there's a way of organizing what's happening in this chapter. And I'll just erase this and just show you this again. And you should create this drawing and just make it really big, and you can fill it in. And so what it's like is, here's a target cell membrane. And so most of these stories are going to be starting with a receptor on the cell membrane. And our first box are the receptor tyrosine kinases. And there are lots of different types of receptors, so we'll put some other ones along here. Of the receptor tyrosine kinases, there are 58 in us, and so we've learned about some. And so the first one is PDGF, and that's the one we know the most about. And we know the signaling molecule with the receptor, the signal transduction pathway, and the cell response. And we know a lot about it, because we know along that cell, all along the signal transduction pathway is RAS. So we learned a lot about it. And our book teaches us most about this. And it's because these often include growth signals. And so as a result, lots of mutations can happen in the genes for the proteins on this pathway, these pathways, that can cause cancer, like RAS. And ERB, a receptor, like that. And the PDGF story, but the one with cancer, right? Like that. And so that's the first one, and that's the one we'll know the most about. And this MTOR story fits on that, like that. What's the other one you mentioned, Ava?
The PI3 AKT.
Yeah, yep, okay. And so I'll just show you that next to this, we'll talk about another receptor, and it's called G protein coupled receptors. And there are hundreds of those in humans. Hundreds and hundreds of those in humans, and it's a really important story. Our book talks less about G protein coupled receptors than receptor tyrosine kinases, and we're learning less because this is a course on molecular mechanisms of cancer. And G protein coupled receptors aren't very often involved in growth. They are a little bit, but usually different stories. Really important stories. I think I mentioned this, just to mention it again if I didn't. It's estimated that 70% of all medicines, all pharmaceuticals, function by affecting a G protein coupled receptor pathway. just hugely significant. So it's really important to be aware of that both in our normal health and our disease states, these other receptor pathways that we'll talk about are just exceedingly important. And there are orthologs of these in every vertebrate, and then orthologs of many of these in every animal. So we could go to fruit flies and learn a lot about these different pathways. So just to mention that, and then we'll put a few more on here too. And I did that on Tuesday, and it's pretty clogged up, but it's really good to spread that learning out and make it physical and make flashcards that go on this. So your question would take us to receptor tyrosine kinases, like that, right, like that. And so that's a really good way to help you organize your learning, is to physically spread it out and say, oh, MTOR and PI3 kinase and phospholipase C and so forth, those are all back here. And they're all stories about signal transduction in these. So these receptors activate a signal transduction pathway like RAS. The same receptor can activate different pathways depending on the cell and the context, and sometimes it can activate multiple pathways. And so in the Tuesday lecture, I say there are really 3 of these that we want to learn some about. So 3 different signaling stories. And the first one is the RAS pathway, like that. And then we can go through these, but we'll know the most about the RAS pathway. And then the second one is, instead of RAS, it's not RAS, we're going to activate a different kind of kinase, and it's going to phosphorylate a phospholipid in the cell membrane called phosphatidylinositol, or it's easiest just to remember PIP. And it's going to phosphorylate it. Phosphatidylinositol phosphate usually has 2 phosphates already, and we're going to phosphorylate it with this kinase to make it 3. And so that's a different signal transduction pathway. Instead of activating RAS, activate this kinase. And then this kinase covalently modifies that odd phospholipid, so we talk a little bit about phospholipids, like that. And that phospholipid goes on to activate a really famous a kinase called AKT. And so AKT is a word I think would be good to learn about in the for the exam. I could ask a question about AKT because it's something that's talked a lot about in chapter 17, even though we don't talk about it very much. This pathway is one we've kind of emphasized because we didn't look at it carefully until chapter 17, but AKT is what phosphorylates FOXO, transcription factor. So we kind of already have an interest in this pathway. And it's a receptor tyrosine kinase that activates this pathway, AKT, and regulating FOXO, and AKT regulates a lot of other stuff too. These pathways can get complicated because they can have branches, and there are branch points, and for us, what we'd just like to know in general is, and just be ready for this, is that when a cell is stimulated to go through the cell cycle, so that's the goal of this RAS pathway in the learning we're doing. RAS can do other things, but it's really typically a growth pathway. There'll be another pathway that's activated that will be like chapter 19, a cell death pathway. And in some cases it stimulates, encourages cell death, sometimes it inhibits it. And that's because it's just considered, it can interpreted that anytime a cell is growing and dividing, it should be thinking about programmed cell death. Because programmed cell death is one way we can eliminate cells that are precancerous cells that are getting out of line. It's just be like, if you're getting out of line, we don't need we don't need you. So So we'll see a little about that, and that's happening in these kind of pathways, okay? So in this pathway is AKT leading to FOXO, great. And another branch point in this brings up the other protein that Ava mentioned, MTOR. And MTOR is a branch point that branches off the same thing that typically stimulates FOXO. And when you see the figure in the book, you're like, whoa, that's a blizzard of stuff happening there. So I would say wouldn't expect you to go there, right? I just don't think anybody's going to be able to learn it in that much detail. There's a lot of figures in chapter 17 where there's a lot of signal transduction story, and probably the most, with the one with the most, is that one that includes the phosphorylation of PIP2 to PIP3, and then activation of AKT kinase that will do some of the FOXO and also affect mTOR, like that. And I would say, even though we mentioned it now in class, I would say, yeah, mTOR is a very famous one. Let's say you don't need to know about it, but let's just say that mTOR stands for target of rapamycin. Rapamycin is an immunosuppression drug. So people that get transplants, like a kidney transplant, like that, organ transplant, it used to be that doctors had to find really good matches, like a sibling, to get a kidney, or they'd reject it. Now we can do that with a little broader ability, because people who get transplants have to take immunosuppressive drugs for the rest of their life to reduce the likelihood that they reject it. And so people are probably a little bit familiar with that idea just out in regular, world. Oh, you might have a transplant. If you do, probably have to take immunosuppressive drugs, and people are doing that. And you probably know something about that, because in COVID, we hear, this is dangerous for people who are immunosuppressed. And they could be immunosuppressed for a variety of reasons, including they're taking a medicine to try to reduce their activity of the immune system, like that. Okay, so just to mention this, because here's the deal with rapamycin. Rapamycin is a drug that was developed 20 years ago, and when they were testing it in mice to see if it was dangerous, it seems really safe. But one thing they noticed is that mice that took rapamycin lived longer. And it slows down their rate of aging. And humans have not been taking rapamycin quite long enough yet to know if it's true in humans. But the reason that our book would bring up mTOR is because it's just like, oh my God. And so mTOR is a partner on a kind of branch pathway with FOXO regulating aging. And mTOR is involved in stimulating growth in growth pathways like that. And it does it by activating ribosomes. More ribosome activity, make more proteins, get more stuff done, grow like that. So again, even though we mentioned it, I would steer clear of it just because it's in pretty complicated part of our book. AKT though comes up, and the book's describing it, and it does phosphorylate FOXO. So I think AKT would come out of that, and that's on this second pathway. So there's a little bit to learn on that second pathway, mostly the kinase phosphorylating this, and a little bit about that phospholipid, because in the third pathway that we want to learn something about for receptor tyrosine kinases, the third pathway involves activating an enzyme that cuts this PIP2, cuts it into two pieces. And that figure is no longer in our book. So it was in an earlier edition, and so I use it because it's really a famous story in another pathway regulated by receptor tyrosine kinases. So that's where all this comes up. See, and is it about G protein coupled receptors or some of these other receptors? Nope. Now, we're kind of becoming experts at receptor tyrosine kinases. We're going to learn a little bit about some of the receptors and then some pathways in detail, especially the RAS pathway. And just in chapter 17, there's a little bit of this pathway. And what we get out of that is really the PIP2 to PIP3 story and how that activates AKT. AKT phosphorylates FOXO. And MTOR is in there, but we're not worried about MTOR, even though it's like, oh my God, right? There's a lot of anti-aging research going on that's really fascinating. And then this third one, even less, but there are figures that I talked about in the second, or in Tuesday's lecture, that are just significant, and they involve what are called second messengers. So some signal transduction pathways activate what are called second messengers, and the idea is that the signaling molecule is the first messenger. It stays outside of the cell, and the signal transduction pathways activate secondary messengers or second messengers, and you probably learned a list of those in intravitreal. So they include things like calcium, cyclic AMP, or for us, these two pieces that come out. One is called inositol phosphate, and or IP, we could just call it, and DAG when it gets cut in two. So that's in that Tuesday lecture, and just kind of a little package to show you that. Sometimes the same receptor, depending on the cell it's in, can activate these different pathways. Lots of times they're activating multiple pathways. A growth factor activates both the FOXO and the MTOR, so we get a lot of branches and a lot of crosstalk. The chapter ends with the book showing examples in receptor tyrosine kinases of that kind of crosstalk and feedback and integration. And we talked a little bit about that. The two terms we want to get, general terms for signal transduction, are that signal transduction pathways involve amplification. So just a few signaling molecules can lead to a strong cellular response because the signal transduction pathway will involve amplification. And then the second thing is what people call integration. And there are, comparable words for integration. A really good one is checks and balances or crosstalk. Sometimes multiple receptor pathways are activated and they are integrated. They are feeding off of each other. Sometimes crosstalk will be a positive feedback loop, increasing the response, sometimes canceling it, like that. And so that's why these are so complicated. And then that they just can do different things in different cell types, like that. So amplification and integration, which means crosstalk, feedback, that's great. Yep. So that's where that fits in, is into that drawing, like that. And it's really good to be able to turn your mind to that and be able to go like, well, what do I know about receptor tyrosine kinases? Like that. Yep. Other questions people have about anything at all? Yeah.
I have a question. You were talking about the ERB protein and it losing a part of itself. It was when you were doing it was based on that drawing. I just didn't understand what part when it went from the EGF binding to the tyrosine kinase domain and then becoming the protein. Is it losing the EGF?
Yeah, so let's just take a look at, so it's a story about a receptor tyrosine kinase. And so here's that RAS pathway for receptor tyrosine kinases. And we can just draw on here. Here's a receptor, and these cell membrane receptors, you really typically have these three parts or domains. So a part of a protein is called a domain. You can think about it like us, right? I'm one protein. I have a head domain, I have a hand domain, I have a leg domain. We talked about that, not about receptors, but about transcription factors. And the transcription factors, which is a different story, right? The transcription factors will have a DNA binding domain and a transacting domain. Different part of the same receptor. The estrogen receptor would have that third ligand binding domain. So domain just means a functional part of a protein. And so these guys on the cell membrane would be receptors that would be said to refer to have three domains. One protein, really three domains. And so the first one is called the ligand binding domain. And so, that's got to have a binding pocket that the ligand binds to. So the PDGF receptor has to have a binding site for PDGF. It's got to also do something where it communicates the presence by changing its shape, like that. When PDGF binds, the PDGF receptor changes its shape. And it's a little like a pair of pliers where, the head of the pliers closes on the PDGF, and that causes the handles to change shape. So the PDGF presence is communicated by binding to the PDGF domain and then affecting the other part of the protein. And that's just interesting because when Riley talks on Tuesday, if you think about it, it's really puzzling and surprising that they can mix and match these parts, because that's what he's going to talk about. He's going to say we took the coding region for the ligand to bind domain of one gene and we just hooked it on up to another one. And so they do have some struggles in mixing and matching parts of these genes that code for receptors to make a CAR, a chimeric antigen receptor, a synthetic receptor that functions. So that's the first one. That would also be referred to as the extracellular domain because it's outside of the cell. So just a really general name, but those are both the same, ligand binding domain, extracellular domain. And then the second one is a little bit boring, and it's just called the transmembrane domain. So it's the one that spans the cell membrane. It's got a lot of hydrophobic amino acids in it like that. And so I should be getting that written down. So first one is the ligand binding domain of the extracellular domain. Second one would be the transmembrane domain. These receptors are called transmembrane receptors because they span the cell membrane. And then the third one, it would typically have different names depending on the receptor. The first generic name would be, that's the intracellular domain. That's inside the cell, that's in the cytoplasm. And then instead of having like a functional name, the receptors would vary depending on it. That intracellular domain varies. So for example, this one, the intracellular domain for receptor tyrosine kinases, people would say, that's a tyrosine kinase. It's an enzyme, and that's how the receptor functions. When PDGF binds to the extracellular ligand binding domain, its presence is communicated to the intracellular domain, and the intracellular domain is a tyrosine kinase, and it activates in it and starts to phosphorylate. In the case of these receptor tyrosine kinases, the target for the phosphorylation is the receptor itself. And so they're said to autophosphorylate. So you can see that. Here's the growth factor, PDGF, binding the PDGF receptor, activating it. It's activated the kinase activity in the intracellular domain, and it's phosphorylated like this, and that allows this to bind and activate the rest. So when the receptor phosphorylates itself, it's on, and it activates downstream pathways. And that's true for each one of those three categories that we talked about. Is the receptor is activated by autophosphorylation, and it binds to something else. The phosphorylated form binds to something else. So we have those three parts, and those would be represented in the gene, right? So if we go over here, and we say, what's the gene for a receptor tyrosine kinase look like? We could just say, well, our favorite is the PDGF receptor. And so here's the transcription unit and the promoter and the enhancer like that, and it's going to get made in the bone marrow, so it's going to have a transcription factor that binds in the bone marrow and activates it. Within the transcription unit, that's the business part, that's going to make the product, and in this case, it's a coding gene, codes for a protein, the PDGF receptor. So this is the business part of it. It's the coding region. The coding region is made up of these triplet letter codons, each coding for one amino acid in the ribosome. And on that, we could just map it, because it turns out you can just map these domains right onto it. So here's the extracellular domain, the ligand binding domain. Here is the transmembrane domain. And here is the intercellular domain, or in this case, it's a tyrosine kinase domain, like that. And again, when these get made into a protein, they all function together to change shape together. And it's surprising that you can manipulate them and get some combinations. In this case, instead of talking about PDGF receptor, the question is about a different receptor that's normally now called the EGF receptor. And our book talks about that, and also talks about its original name, which was ERB-B. You don't need to know it, but that stands for erythroblastoma, and it was originally identified as an oncogenic retrovirus. So back in the 1970s, this normal gene, EGF receptor, that's used in our body to regulate growth, stands for epidermal growth factor. The receptor binds the epidermal growth factor, EGF, that's used a lot. And so it's just one of those. And in that retrovirus, and then more recently, in a variety of just cancers in humans, what's found is the damage is that part of this coding region in the extracellular ligand binding domain just got deleted. And so it makes a protein, and the protein is a shortened version of the regular protein. And so it's said to be truncated, and that's not really a science word, it's this English language word that means to shorten, cut short, like that. Okay, so we're going to make a truncated product, and it just happens that for this one, and really not so much the other receptor tyrosine kinases, that if you make one without the ligand binding domain, it's just activated. It's lost the need to have a ligand to just
In
activate.
ERB.
Yeah, in ERB. And, you know, like if you cut the front half of the pliers off, well, they're just no good. But so that's what typically happens with the other receptor tyrosine kinases. If you delete the front part of the coding region, they're just junk. This one, it just happens that if you delete it just right, you can leave the intracellular domain expressed and active. And so that's that. You held up that image because it's it's not from our textbook, of course, but it's just a nice figure. I have it on here somewhere. And it's just trying to show that here, right? This is the normal ERB-B protein EGF receptor, and it normally binds to EGF, looking good, doing its job. And then this is the, you know, it's kind of artistic rendering that I think is kind of pleasant because it's like, ooh, ouch. It's just a truncated, shortened version of it, and that happened in the DNA as a type of mutation, just deleted it, lost it, don't have it. So you make this shortened version, and it just happens that it is always on.
And the ERB protein caused that?
Your B protein is that. It's the product, not the cause, but the product. Yeah. What caused it was some kind of DNA damage. So we're learning in this class that there can be a variety of types of mutations. In intro bio, you really learned, oh, there could be these point mutations where you could get a single nucleotide letter, like maybe an A changing to a G, and that could cause trouble. And it does in what's our favorite example of a oncogene, a gene that can cause cancer that is damaged by point mutations. Our favorite example of that is RAS, because that's where the RAS protein has to turn itself off by hydrolyzing the GTP. And that means the RAS protein has amino acids that fit into the active site of an enzyme activity. And the RAS gene can get point mutations, random mutations, changing specific amino acids that would be lining that domain and damage it. And then the RAS fails to turn itself off. That's different than the kind of mutation that the woman has with the skin cancer. That's not a point mutation, and it's no problem in the coding region. She makes normal PDGF. The problem is in the regulatory region. The 6 regulatory region got exchanged by a mutation, a different kind of mutation, a chromosomal translocation. Yep. And so this is a different kind of mutation too, where you just delete it, you just lost a chunk of, and it's got to be the coding region. It's not the regulatory region. It's not an issue of where, when, and how much. It's what the product is, and it just makes this defective product that's always activated like that. Doesn't need the ligand, okay? That's where we're at. We talked about some other mutations. Yeah,
So is ERB specifically the name of the mutated version? And
Yeah.
then normally it's the EGF receptor?
yeah, the way to remember that is just that these oncogenic retroviruses were studied back in the 1970s where nobody knew anything about normal genes. And so here's this wicked, wicked gene, and it could cause cancer. And it wasn't in humans, it was in some other animal they found it in, you know, and it would just cause cancer when it infected it. And they mapped it out, and they're like, whoa, it's one of the animal's normal genes that's causing the trouble. And then in the 1980s, they found out, oh, it's because the normal gene's gotten shortened, and it's just active, like that. And then in the 1990s, they're like, hey, we know more about that regular gene now. The regular gene codes for a receptor, that's a receptor tyrosine kinase, and you know, we already knew about this hormone that it binds to, this growth factor it binds to, EGF. So we should call this the EGF receptor. But you know, that's the deal. But it's nice in this case just to keep track of ERB, because people like to call the damaged form ERB just because they cry when they say it, because it's just so sad and evil, tragically evil, you know, because you're just like, dang it. You know, and that's why, you know, Joe asked this question is, what's going on with that? And it's just, in this case, you could just make a shortened version of it, and it's lost its self-control as a protein. It no longer needs a ligand. Doesn't even need the ligand binding domain. It's just active. And who's active? The autophosphorylating tyrosine kinase intracellular domain is there, and it's just like, oh yeah, go, 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 like that, right? Okay, so that's the context for that. Yep, yeah. And where it comes up is this cetuximab. So there's an antibody, a protein that scientists have found, a company, you know, built this, and it's a product, and it's a pharmaceutical, it's a drug that you would inject, and it's a chemotherapeutic agent, and it binds, whoop, where we go and lose that? It binds to this truncated form, and it activates it, turns it off, like that. And so it was approved 14 years ago to use in cancers that are caused by that. You wouldn't use cetuximab in any cancer. It would not help at all, right? It only works on that. And it's a monoclonal antibody. They talk about that. You know, we don't need to learn about it, but you know, people learned a lot, heard a lot about antibodies with the pandemic, right? They're proteins made in our body, and we talked a little bit about them in this class, but it's not a immunology class. But just think of it as a kind of a natural product that could be used as a drug to stop that activity. Yep.
So is it that like cetuximab, because then that ties into RAS, so is it that it binds to RAS and is able to deactivate it? Is that what that's doing?
No, but that's a good question. And I talk about it in the recording on Tuesday, and it works like this. And it's just really satisfying. And it's like that Tamoxifen story. I even talk about that on Tuesday, right next to this. So just remember the Tamoxifen story is, it's a drug that you can use on breast cancers that are ER positive, but not ER negative, useless. So this is a story like that, and it's in chapter 20 in our book. The book tells some stories. So this cetuximab comes up in chapter 20 more, but we want to learn about it in chapter 17 because it's a chapter 17 example, really. And so we're picking and choosing which ones to talk about in class. And when you read chapter 20, it'll be nice because there are a few different examples that you'll be like, nice, I know this and I can take this up a notch, right? We did that with chapter 9. Our very first thing in the class is to watch the videos about transcription. That's chapter 9. You know, so we want to prepare for chapter 20 by talking about something. So this is from chapter 20, but we need to know it now from the lecture. And this cetuximab stops that. And then here's the thing, it's like the Tamoxifen story, but it's just really satisfying to hear the logic, the cause and effect relationship, and the common sense of it, okay? So, and I do this in the lecture, And so here's this setrusumab, and it just talks about it, it's like a epidermal growth factor receptor inhibitor, great. And then it's down here that you just go, oh my God. So in July of 2009, so it's going to talk about early days in these kind of really targeted chemotherapeutic strategies. Tamoxifen has been around for a long time, but they really didn't know too much about tamoxifen. One reason that tamoxifen is talked about a lot is because they used to use it on every breast cancer, and it didn't work on a lot, right? And they had to figure out more to say, oh, now we understand what tamoxifen is. It's an antagonist of estrogen. So it's only going to work in a breast cancer that's got estrogen receptor dependent, right? So that's older. They're learning more. But this is some of the first stuff, and it's just 14 years ago. So they say the FDA approved setrusumab for treatment of colon cancer. Why colon cancer? Because people find that ERB mutation occurs kind of frequently in colon cancer. Okay. Now, what does that mean? What that means is that normal colons, our normal healthy colons, use the EGF receptor as part of their normal health. And if that gets mutated, it can cause trouble. In another tissue, if not using the normal EGF receptor, let's say the brain doesn't. Well, if it gets mutated in the brain, it's not even being used. So not going to be a big problem. And so we see that, right? The one with skin cancer, PDGF receptor, it's there, the skin is ready to respond to PDGF. If you misexpress it, autocrine response, like that, right? So that's one thing, is it says implicitly, oh, the EGF receptor is used in the colon normally, and if it gets one of these deletions where it happens to lose that part of the coding region, it's growing out of control. And that happens in cancer, like that. And then it says that also happens in certain kinds of lung cancer, just certain kinds, and certain kinds of head and neck cancer. Again, it's because the normal biology uses that system, and if you delete it, oh shoot, right? Okay, so they approved it first to treat it with colon cancer, and then they say that FDA treated it and said you can use it, but they said only if it has wild type RAS. Since setrusumab had little or no effect on colorectal tumors that had RAS mutations. And just to show you that idea, where is it? To find the pathway. So this is that RAS pathway, and we're saying, oh, that ERB is growing out of control. We could slow it down. That would help. And the FDA says, yeah, you can use your product, you can sell it, use your product, but not if the patient has mutations in RAS, because RAS is downstream of ERB. And in some patients, because cancer progresses by getting multiple mutations, gets worse and worse. Some patients, they have a mutation creating the ERB, out of control, and then further out of control by having a mutation with RAS. And if you have both of those, if you slow this down, it's not going to help the patient very much, maybe not at all, because they're just going to have RAS raging on, like that. So the FDA says, kind of like what we learned about tamoxifen and some other examples, and you can't just use this. You can't just use setrusumab. And you can't even use it if you say, oh, they have this ERB mutation, should help, because it won't help if the patient also has mutations in RAS. So see that? That's the physical, right? And it's all laid out physical, right? So setrusumab bind to RAS? Nope. Setrusumab is an antibody. They stay outside of cells. They never get inside a cell like that. So nope, doesn't bind to RAS. It binds what you would say upstream. In this pathway, we have sort of early steps and later steps, upstream and downstream, right? Like that. So it's just really satisfying to see that. Oh, now we have to find our little FDA page again. It's just really satisfying to see this logic, right, the FDA is using. So then it says this. It says, you can only use your new drug, setrusumab, you can only use it if you prove that patient has no mutations in RAS. So then the company's like, oh my gosh, we have to be able to test patients to test their RAS gene easily. So then it says, we developed a genetic test to guide treatment of cancer. And it says this was the first example of that. And like I said, tamoxifen, there were a couple other things that were used earlier than that, but it's just been the last 14 years where scientists and doctors are really grinding on this idea of pharmaceuticals that are very specifically targeted to specific problems, specific mutations in specific kinds of cancer.
Sounds expensive.
Well, so that's a good thing, is definitely expensive, but it's one of these things like that I mentioned with that CAR T cell therapy. They sometimes talk about that when they talked about look at the Wright brothers, they invented flying. And there it would have been expensive to try to figure out how to fly a plane or carry some Amazon packages on a plane. But then once you get rolling on that, right, prices come down, right, things get streamlined, scientists understand it more, you get some competitors making a product, and the prices start coming down. So yeah, early days in these kind of treatments are ridiculously expensive and prohibitive, and the hope is that will drive, the drug companies forward to make competitive products and blah, 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 other treatments. Yep.
Usually until like the patent runs out, it's pretty expensive, but like, I work as a pharmacy tech, like in a pharmacy, so
Yep.
like dealing with like insurances, like a lot of time like insurances don't wanna cover
Sure.
something that's
Yep.
like
Oh, yeah.
expensive.
Yeah, absolutely, right? Yes. So just to read this last sentence, so this was the first genetic test to guide treatment of cancer. The doctors had to test the patients for wild type RAS, make sure they didn't have mutations in RAS, and then say, okay, we can use setrusumab on this patient, like that. And it says in July 2012, so 2009 they approved the drug, but they were like, we can't really use it yet based on the approval. So they scrambled and 3 years later got this drug test. And the drug test is a PCR test. You get some of the patient's blood and or tissue, biopsy from the tumor rather, and you test it with PCR to see what's going on with the RAS gene, right? And that's just a really satisfying paragraph. And it takes it up a step from the tamoxifen story. We're solid on that. This is a different kind of cancer story. It's really a chapter 20 story, but we're dealing with it now because we're leaning in on little pieces as foreshadowing where our course is going. And why? Why are we learning about the receptor tyrosine kinases, right? These are reasons why, like that, yeah? Yeah, so makes sense, right? Joe and everybody, right? Nope, the antibody does not bind RAS, doesn't regulate RAS, it regulates the ERB truncated EGF receptor. And then we bring up RAS because it's downstream, and we wonder what's going on with the RAS, yeah? Yeah. We're doing great. Ooh, did I hand out a sign-up sheet? That's out there. Okay, good. And we'll have a concept chat in a minute. Other people have questions? Let me show you a couple of slides. There isn't very much more new material to talk about in chapter 17, but on Tuesday I found an old slide stuck in from last year that I hadn't really updated to what we're talking about. So let me just scroll and find a couple of slides to show you what happens in the end of chapter 17 is, this is one that I didn't change. This is from our Tuesday's lecture, so we have this. And this is the way the receptors are organized that we want to learn about. So lots about receptor tyrosine kinases, some about nuclear receptors, right? That's a really different category. And then a little bit about androgen receptors. And then there are these 4 others that are very different kinds of receptors and stories, but the key thing for us is each one of these 4 regulates a specific transcription factor. And we really like that. We really like to see the transcription factors getting regulated. So as a result, we just go boom, boom, boom. And it's really, I got through this one on Tuesday, and we just need to talk about those 3 like that. And I'll just show you that as we talk about those 3, we are adding to this list of the dynamic story of transcription regulation. And in the lectures, we talked about Krebs and STAT is one of those. I talked about that in the JAK-STAT pathway. And those fit in here. They're regulated by phosphorylation. So there are transcription factors that get switched on and off. Those get switched on by phosphorylation. and through a signaling pathway, like that. Okay? And we just need to add a couple more down there on Tuesday. But they're important, and so we do have a little more learning to do on Tuesday for our first exam. But then you can see lots of questions trying to piece this together. So there's probably about 20 minutes of Riley on Tuesday, and then 10 minutes of me talking about these that you can see now. I can just show you the list again, and that's on Tuesday, so you know these are their favorites. And each one of them is one figure in a little section. So go to this list because we need to learn those as examples. And each of these would be like a 2-point question like that on the exam. Just like, what's that about? Yep. Okay. And so I just want to show you that because that provides a sort of navigation for that chapter 17. There are some other pathways the book talks about, like NF-kappa B and stuff like that. And those are much less important. And, again, we would rank them, the way I've ranked them. We just need to know receptor tyrosine kinase. Probably half of our learning needs to be that, more than half. And the 3 different pathways, and like that. And then nuclear receptors, wow, those are pretty cool. And then these others are kind of boom, boom, boom to fill out what the book is trying to teach us. Okay, so let's do a concept chat, Yahoo, and then we'll keep talking a little bit more. And so try to find one person, and let's see, we have an uneven number of people, so there'll be one group of 3, but other groups of 2 is good, because we want everybody to get a chance to talk. So that's why we're going with groups of 2. And fantastic. And so there could be one group of 3. Some people thought it was a snow day today. No, they were realistic. They were coming from Skowhegan. All right, so just talk about 3 things that you each thought were important today.
I don't need it, but I think the R might be receptor. But it takes the place of the EGF binding site.
So we'll just do this another minute, and then we'll check in. All right, so Amal, what is one thing that your group thought was important today?
We were talking about the drug treatment that they use, like the chemotherapy, and how it targets the ERB.
Yep. Yeah. Yeah. So that ERB story, and its mutated form that's found in some cancers, and it was originally discovered in an oncogenic retrovirus. So scientists have known about it for a long time and trying to understand it. And so it's one of the things about those that were studied in the 1970s that they tend to be, scientists know more about them. And so they've seen, there are certain cancers that have this ERB mutation. And so, probably in 1995, a company started trying to make antibody that would bind to that mutated form and block it.
So I just have a quick question about retroviruses and oncogenic. So in order for some of these cancers, do you have to be infected by the retrovirus?
No. And so remember the way we think about it, and again, you don't need to learn the years and the names, but the way we think about it is our author, when he was your age, was in grad school in Wisconsin. And they were studying oncogenic retroviruses in Wisconsin and other places. And they were studying the one that had RAS in it. They didn't know. No one knew what RAS was. What causes the cancer? And in that lab there, they discovered it was one of the animal's own genes that was the cancer-causing gene. So we talked about that these oncogenic retroviruses had accidentally picked up a host gene, and it was that host gene that was defective and caused cancer. And the way that happens for RAS is it picks up a host gene and then it gets some point mutations. So it provides this out-of-control RAS. In the case of MYC, it's the normal MYC, but it picks up way too much expression. And remember, MYC is just carefully regulated. The RNA is unstable, the protein's unstable, it can't translate it, it's got lots and lots of controls on it, but you just make too much of normal MYC. And then for this ERB, it just picked up that EGF receptor, but it just picked up a part of it. And so that's happening back in the 1970s when our author was in Wisconsin. And we like talking about our author because then our author went, after he finished grad school, to Boston, to Harvard, and left the retroviruses behind and looked at normal human cancers, and he tore up the DNA from human tumors, put it in mice, found pieces of the DNA could cause cancer, and found that what we now know as RAS again. And the reason we like to talk about it is because we left the retroviruses behind. They were the foot in the door, and then, So your question is good, because students get confused by that. We keep talking about oncogenic retroviruses and going back to it, but that's just the foot in the door, and all of those are, in our bodies, normal genes that, separate from any retrovirus story, we could eat too many tacos, and we could get a mutation in RAS.
Yeah, so it's just from the physical process of the transcription that leads to those.
Viruses just make a lot of mistakes.
Right.
And so when you're talking about viral infections, you're playing with danger. And in the case of these retroviruses, their replication cycle, we could draw on the test and describe it. It includes integrating it into the host chromosome and then transcribing it to make more RNAs to go out to be more viruses. And that's just a dangerous game because you could land right next to an oncogene like the EGF receptor and accidentally pick up part of it. And we said, is that going to happen with COVID? And everybody's like, no, because COVID's replication cycle does not involve integrating into the chromosome. So COVID's not going to become, COVID cancer virus. It goes back to that same Tamoxifen or Cetuximab logic. You're just like, nope. And people out in the general public, they're like, wow, you scientists are sure confident. And you're like, yup. The car's out of gas. We don't need to check the tires. like that. It just, like that, but we're also skeptical and welcome and forward-looking, so we're trying to make more discoveries. Maybe we find some details, and you're like, wow, we're surprised, we love that, but there's some times where you're just like, yeah, no, and that's a bunch of molecular biology where you're just like, yeah, it's the way it is. Let's see, did you have a question?
If that mutation occurs in a germline cell, specifically for RAS, is that going to be passed down to the next generation, or is that a terminating mutation?
Yeah, that's a good question. So we go to this idea that in some families, they have certain cancers running in their family, they'd say. Like certain kind of skin cancer runs in so-and-so's family. Just a higher frequency of people in that family get that cancer. What's going on with that is they're inheriting some gene to their offspring down the generations that's got a mutation that makes them vulnerable to that kind of cancer. And so if they get more mutations, they're kind of set up to be, one step away from getting that kind of cancer. So you use the word germline. Germline just means the cells that can make sperm and egg. Those are the cells that will be inherited. And that's just important to think about. If you go to the beach and you get sunburned and you get skin cancer, that's not in your sperm and egg cells. It's not going to be sent on to your children like that. Only mutations that would happen to the germline in our ovary and testes could get sent on like that. So it's interesting to think about that with regard to cancer. There are a lot of cancers that are in cells, the brain cancer, skin cancer. That's not going to harm your future offspring. RAS could be mutated, and it's a point mutation, so there are different kinds of point mutations, and in some cases, you're just weakening it, and so a person could have it and be okay, but suddenly be in real big trouble if they got like an ERB mutation, because then they have that double whammy, with a little bit of RAS out of control, and you're okay, making it through life, but then you get that second one. So yeah, I'm speculating, but I would say, oh yeah, definitely certain RAS mutations could be in the germline and they could be inherited in a family like that. Because when we talk about mutations in this lab, we're usually talking about ones that are really damaging, where you're like, oh my gosh, oh wow, like that. And there could be comparable, similar mutations that would just not be that damaging. And it would make a person really vulnerable every day of their life, to pick that up. Yeah, I'd say that. Yep. Let's see. So we talked to Amal. Let's talk to Catherine also. What is another thing that you guys talked about?
We talked about the three sections, or the three domains of the receptor, and how that corresponds to three physical sections of the gene.
Yeah. And that's really common in proteins, is that they'll have, kind of a way of describing their physical shape based on the different functions they have to do. And again, it's kind of like our body, where I said, I have a head domain for chewing and hearing, like that. That's what that domain's doing, and it's different than my hand domain. And I'm one protein with multiple domains like that. Got the leg domains to, move around. Yep. And then Brooklyn, what's one thing more?
We talked about how it was interesting that 70% of pharmaceuticals target the G protein, or the G.
Yeah, it's kind of a thing to learn. G protein coupled receptors. Yeah. So receptors that work through a different kind of G protein, not RAS, but another kind. Yeah.
And we looked at figures that showed all of those pathways moving throughout.
Yeah.
She found on the internet.
Yeah, and it's great. And we ran out of time, but I think this weekend studying, we've shown, draw these things out and tape a piece of paper together, find some colored markers, a couple of pencils. I have a bunch you can borrow. You don't have to go and buy stuff, if you wanted to borrow some markers to make these and make flashcards that lay on those to navigate from here to there, where you're like, oh, nope, I'm learning about this over here. Now I need to learn this piece because I don't learn it yet. Yep, okay. So we'll meet with Riley on Tuesday, then me a little bit more on Tuesday that I tried to talk about, and then we'll have our awesome exam on Thursday.